Really? Too deep one? Oh, some big old copepods in there. So I like to tell people I'm a disturbance ecologist, not a disturbed ecologist. It'd be really interesting to know if there's any distribution between the shallow and the deep. Probably the thing I enjoy most about my job at Plattsburgh is getting projects going and then getting students involved in projects. I think I knew as a child that I was interested in water. I was the kid in the ditch when it rained, playing in the water. As I finished my undergraduate and ended up in graduate school, I realized it's the biota in water that fascinate me the most. Streams and insects and rivers, plankton and fish and other things in lakes. My job is very fulfilling at SUNY Plattsburgh, and I always say I really like my job. I get to do a lot of different things. We have a lot of projects going on in the Research Institute. One of the main ones is the long-term monitoring program, which we've been supporting at Plattsburgh for decades. We process all the biological samples of plankton from the lake and get that data together for the monitoring program. And then we look at that data and help try to understand the patterns and what's going on and what's changing in the lake. So today, we're doing diel vertical migration. So we're doing multiple depths. They're doing 5-meter intervals with 3 different net types. And we're getting the basically plankton that live in the different strata of the lake from 0 down to 50 meters. The whole ecosystem depends on this vertically structured thermal structure in the lake. So if it changes, for instance, it changes with climate change or storm events disrupt it, you're messing around with all of these species and their evolution and their biology to orient toward this vertical structure. So it's pretty important. We're at 15, going to 10. Right now, sort of the biological responses that we've seen in Lake Champlain, especially in the plankton, are a story of invasive species. We had the early 90s onset of zebra mussels in the lake. They had a huge change in the lake's biota. Our mysid shrimp populations, which is one of the largest predators in the plankton in the lake, crashed in the 90s, 3 years after the zebra mussels invaded. And then most recently, we've had 2 invaders invade Lake Champlain. It was a student in my lab that discovered the spiny water flea in 2014. It was a different student in my lab that discovered the fishhook water flea in 2018. Those were not wonderful moments, but students were there going, oh my God, this thing just invaded the lake. And then they come to me, and then we go down to the lab, and then we all get upset. Getting students involved in this work is important because they're going to be the stewards of the future of what we do in Lake Champlain.
You didn't see just
3, 2, 1.
I'd say directions for the future for me as a scientist will probably be to make sure our monitoring program is on solid, stable ground and that we're contributing to that into the future. We have a new study on microplastics in the lake. We're going to survey the entire lake, all the monitoring samples, and look to see where plastics are, try to get a handle on the distribution of plastics lake-wide, which is a huge issue, and it's an environmental issue that I think we need to do a little more to try to figure out and then sort of tackle that problem in all water bodies, including Lake Champlain. The lake is a divide between New York and Vermont, but it also brings us together. We share this lake together, everyone who lives around it. Quebec, New York, Vermont, we all share Lake Champlain. And in many ways, I've been able to work with a lot of people over the last 20+ years who share the values of preserving and conserving what this lake is, keeping it clean, keeping it healthy, and leaving it for future generations. Yeah, it's a dream job, I guess you would say.